In this lesson, we will learn about ecological genomics. So first of all, outline, we will see introduction to bioinformatics, then what is genome annotation and genome evolution. So in this figure, this is the genome, which is having the complete, all of the chromosomes carrying all the genes. So if we take one chromosome and open it and we see this is the DNA sequence, and this DNA sequence is having genes that actually code for a particular protein. The genes actually carry the instructions for making protein. So there are different proteins. The protein can be of hair protein, can be nail, can be keratin, any sort of protein. And these proteins actually they do not act alone, but they act in complexes and perform different functions wherever the function is required in body. So, if we talk about biology, and we will like to combine the biology and informational science. So, we can say that after the Human Genome Project, HGP actually stands for Human Genome Project. So, once Human Genome Project has been launched, so there's actually a very drastic change in how particular scientists, especially biological scientists, they do research in that field and they practice biology, and they just the domain changes, and biology turns out to be an informational science, which is not only biology but it carries all other disciplines. So it actually has Human Genome Project give an idea that it is having digital genome and environmental signals. They both play a very important role in the genome, in the genetic makeup of an individual. So digital genome is actually carrying all of the genetic makeup of an individual, and it is with the help of different computer softwares and techniques. It is helpful. They apply on the genome, and it is helpful in identifying different disorders. And then they talk about the environmental signal that they are very important in formulating any genome of an individual. That genes of an individual are affected by the environment in which it's present and the environmental signals. So as a result of this, all of this information, we came to know that biology is actually a cross-disciplinary science. It's not only biology. In fact, it is a mixture of the different sciences that actually help in understanding of Human Genome Project. So we are having mathematical science, we are having computer science, and then we are having life sciences biology. And when we combine all of these three techniques, we came to know that there's a new domain of biology which is called bioinformatics. So it comes in market. So bioinformatics is actually a combination of different computational and life sciences, like mathematical science, computer science, and life sciences. All of three combined, and then there is a new field which is called bioinformatics. So now the question is how actually bioinformatics is integrated and how we generate data and we use it. So first is that in order to generate like that all the genetic makeup, we get huge data sets. So in order to get the genome or genetic makeup of an individual or to understand it, we need to have a biological background. And once we have a biological background and we get huge data sets of our gene, the genetic makeup, then this is raw data. And in order to use it as a digitalize this data, we need to have an IT background, or IT platform is needed to convert that biological life sciences genetic data into digital format. So once it is converted into digital format, then what message that genetic data is providing? So there must be patterns in that data sets. So different mathematical or softwares are needed. Or there here we are using computer, and then here we are using different mathematical and computer softwares, which will be helpful in understanding the message that biological data is giving. And then we need different resources to integrate that data, like different diseases, and we want to integrate each one, that this is giving this message, this is giving, and then there's any correlation among different genes or any kind of pattern. So this is how this actually all build up is building up towards the bioinformatics. Like first we are having whole genetic data, then with the help of IT computer science we are storing that data, and then this is the raw data. We can't understand any pattern, and also understand the pattern we use different mathematical softwares, and using computers, we apply that software, understand the message that data is giving. And once we understand that, so we use different resources like different tools to integrate that data. Like just collect a whole of the one complete set of data giving information about a particular disorder or disease. We integrate it in one resource, and then for the other we integrate it in another resource. This is how bioinformatics actually works or is developed. So still, what is bioinformatics? So now we can define first, like previously we have in previous slide, we have an understanding that how actually bioinformatics works. Like there's a computer, there's a mathematical, there's a biological, and all integrate. But now we move towards this definition. So what actually is? So this is the field of biology which is specialized in developing hardware and software to store and analyze a huge amount of data generated by life scientists. As explained in previous slide, like life scientists are biologists, so they generate huge amount of data of genes. Then, with the help of hardware and software, using computers and mathematical softwares, we use to store the data, and then we analyze that data. So, if you see, there are various definitions. Then we have two more interesting terms: computational biology and computational molecular biology. So, we will see these in coming slides. But before that, I would like you people to just brainstorm on this, what you get from these words. As I have explained you previously, that computational is all mathematical and biology, mathematical and computer fields combined in the life sciences of biology. So, what do you get an idea that what is the computational biology and what is computational molecular biology? Because being biologists, we know the definition of biology, we know the definition of molecular biology. But when we add the word computational, what idea did you people get? So let's think over it. So now comes what are the key challenges of bioinformatics? So as I've told you that Human Genome Project actually revolutionized the field of biology and the thinking of biologists in the way they were thinking. So actually, it's very different from what biologists were actually working or thinking about in 10 years ago. So the main goal of, because now they have come with a new idea of using computer and maths to understand and store and analyze the biological data. So main goal of bioinformatics was, after that was the few challenges faced by bioinformatics or this field, especially for the biologists, is actually to bridge the gap between technical data production and its use by scientists. Because when the bioinformatics was introduced or this field was in the initial phase, the biologists were not having the background of computer or maths, and computer and math people are not having the background of biology. So they were the key challenge to bridge a gap between both of them, so that the biologists can use computer softwares to better interpret their data, and computer and mathematical people, they understand biological data so that they can apply their softwares and help mm, uh, people in biology to understand the underlying mechanism. So this was actually the main challenge. So now coming to some of the few basic definitions after bioinformatics. This is ecological genomics, which is actually piece to understand the genetic mechanism underlying response of an organism to their natural environments. So then genomics is actually having the complete genome or complete genetic makeup of an individual, and omic actually is using different computer. The omic is actually the softwares or mathematical models. So with the help of them, it's like the understanding the underlying mechanism. Then genome annotation. Genome annotation is actually the process of identifying of functional elements along the sequence of a genome. Like if we commonly say that we are annotating the genome. So actually we are understanding or trying to identify the functional element that is working in the sequence of a genome. It is a sequence because it is very important because the sequence of DNA produces sequence of unknown function. Sequencing, once you sequence whole of the DNA, there might be some sequences for which we don't know the function. So in order to avoid them or to identify those with unknown and the known function, we go for genome annotation. So another definition, the detailed definition, is that it consists of describing the function of the product of a predicted gene. So in silico, of course, in silico means using computer and different softwares. So an example of which bioinformatics software are helpful in annotating a genome, because as told in previous slide, there might be some sequences which are of unknown function. So in order to understand them, what is their function? We do gene annotation. So using computer software. So what are there are three main points of gene annotation. We can use signal sensors. Again, in silico approach using softwares, like we identify data box, start codon, stop codon of that gene, Fourier signal detection, then content sensor that in particular gene of unknown function, we identify GC content or codons. Then similarity detection between proteins from closely related organisms, like if we are having a genome sequence of unknown function. So what we do is that we identify that particular sequence in different species or related species, and then see that is the sequence is same in all of them, and we identify the what is the role. Like for example, in humans, we identify a sequence and we don't know its function. Then we see in the related species or organisms the same sequence and see what function is performing there. So it might be that the function is known in that organism or species. So from that, we infer that it might be performing the same function in humans. So this is the way how we perform gene annotation. Then is the process of genome evolution. It's the process by which a genome changes in structure over time through mutation, horizontal gene transfer, and sexual reproduction. That is the process by which a genome is evolutionary, like evolution. It goes changes from generation to generation. Then computational biology, which I have asked you in previous slide, you might have the answer for that. But so let's see your answers match with this one. So computational biology is actually a branch of biology involving the application of computer and to the understanding and modeling of structure and process of life, because biology is the study of life. So in computational, we simply use computer or different computer softwares or models to understand the processes of life. Then computational molecular biology. So molecular biology is actually detail in which we use experiments to understand the detailed mechanisms. So keeping that in mind, we say that it brings together computational, statistical, experimental, and technological methods in order to further scientific discovery and develop new analytical tools for molecular biology to understand the deep down molecular mechanism. So this is the difference between computational and computational molecular biology, but both they fall under the domain of bioinformatics. So this was the end for today's talk, this lecture. Hope you understand it. Thank you. Allah Hafiz.